Hello everyone. My name is Dr. Munmi Bora, plant virologist, Department of Plant Pathology, Assam Agricultural University, Jorhat, Assam, India. Today we are going to discuss about coronavirus SARS-CoV-2, its infection process, to challenges in vaccine development. This lecture is based on some literature survey and newspaper articles. Let's take a look into the discussion about the virus SARS-CoV-2 causing havoc to this entire world and the pandemic of this century. What we are going to learn today, we will be learning about coronavirus and its origin, coronaviruses that are SARS, MERS and COVID-19, the virus structure and infection process inside host system, epidemiology of SARS-CoV-2, diagnosis, treatment, vaccine development, challenges in vaccine development in case of pandemics, type of vaccines, vaccine development process, and what in news regarding SARS-CoV-2 vaccine development status and the prevention. A coronavirus is a genus of virus, is a kind of common virus that causes an infection in your nose, sinuses or upper throat. They are found in human and animals. Most coronaviruses are not dangerous. They were first identified in the 1960s. The symptoms of most coronaviruses are similar to any other upper respiratory infection, including a runny nose, coughing, and sometimes fever. In most cases, you won't know whether you have a coronavirus or any different cold-causing virus. Generally, we treat this kind of coronavirus infection the same way we treat a cold. But diseases due to coronaviruses such as SARS, that is severe acute respiratory syndrome, MERS, Middle East respiratory syndrome, and recently the COVID-19, that is coronavirus infectious disease 2019, are so dangerous, causing havoc to this entire world. Research suggests that the original source of coronavirus that caused COVID-19 was bats and pangolins. They have acted as intermediates. The COVID-19 was first identified in Wuhan, China during the year 2019. The mutations and natural selection might have taken place either inside pangolins or in humans after transfer from pangolins. Researchers are not sure what caused it. Many people who got the disease early were linked to a large live seafood and animal market in China, which is called wet market. The first cases may have come from animals sold in that market and then spread from person to person. So, effects due to coronavirus historically reported as severe acute respiratory syndrome, SARS, then Middle East respiratory syndrome, MERS, and recently COVID-19, that is coronavirus infectious disease 2019. In 2003, 774 people died from an outbreak of SARS, which is thought to have evolved from infecting bats to civet cats to human in southern China. Then about 58 people have died from MERS outbreak, which first appeared in Saudi Arabia and then in other countries in the Middle East, Africa, Asia, and Europe. Research says COVID-19 is an example of SARS, which is known to transmit from bats to pangolins to human from the local seafood market in Wuhan, China. Why it is in news now during 2020? Actually, after a December 2019 outbreak in China, the World Health Organization in early 2020 identified SARS-CoV-2 as a new type of coronavirus, and the outbreak quickly spread around the world. So, COVID-19 caused by SARS-CoV-2 virus that can trigger a respiratory tract infection spreads the same way other coronaviruses do, mainly through person-to-person contact. Infection ranges from mild to deadly. So let's have a look to the SARS-CoV-2 virus, its structure and infection process inside host. SARS-CoV-2, the abbreviation of which is Severe Acute Respiratory Syndrome Coronavirus 2. These viruses are spherical shaped and they consist of genetic material RNA, which is surrounded by an envelope with mushroom-shaped protein spikes. These protein spikes give the appearance of a crown. Crown in Latin is called as corona, and this is how the virus got its name. The spike protein of novel coronavirus shares 98% sequence identity with spike proteins of SARS coronavirus. SARS-CoV-2 is having 4 structural proteins similar to other coronaviruses. These are S, E, M, and N. That is spike, envelope, membrane, and nucleocapsid proteins. Let's take a look to these proteins. The spike proteins contain a receptor binding domain. The receptor binding domain facilitates the virus entry into target cells by binding with the cellular receptor called angiotensin converting enzyme 2 ACE2, which is found in heart, lungs, kidneys and gastrointestinal tract. Spike proteins itself has 2 functional units. S1 binds to host cell receptor and S2 mediates fusion of viral and cellular membrane. Angiotensin converting enzyme binds to S2 receptor. S2 proteins of novel coronavirus binds to the cell receptor with much higher affinity, that is 10 to 20-fold higher compared to SARS. The stronger bonding affinity partly explains the apparent high human-to-human transmissibility and a faster spread of COVID-19. Next is membrane protein, that is M, which is most abundant on viral surface. It is the central organizer for viral assembly and interact with other structural proteins. Moving into the E proteins or envelope proteins. These are smallest of all structural proteins on viral membrane which plays several roles, most importantly on viral assembly and release. Underneath the spike protein, we find viral enveloped, the outermost layer derived from host cell organelles. It is a fatty layer, and in contact with soap, this layer breaks down, killing the virus. This is the reason hand washing is so important to prevent spread of the virus. Then underneath the envelope there is capsid which encloses the genetic material, that is RNA. Inside this capsid, N protein present. This protein has a major role in viral RNA replication and it inhibits many of host cell defense mechanisms. So once inside, the virus hijacks the cell's reproductive machinery to produce more copies of itself before breaking out of the cell again and killing it in the process. Viral epidemiology is the scientific discipline concerned with the study of incidence and spread of viruses in population over time, and the ultimate goal of which is to devise intervention strategies. So the geographic distribution of COVID-19. The disease have been reported in all the continents except Antarctica and have been steadily rising around the world. Then route of transmission. Transmission mainly via respiratory droplets of an infected person through aerosol in the air. Then reproduction number. So reproduction number means the number of additional cases that likely to result from the initial case. It indicates the average number of people who will contract the virus from an infected person in a population that does not have immunity for the said disease. So you have heard about the herd immunity or vaccination. So herd immunity means the resistance to the spread of a contagious disease within a population that results if a sufficiently high proportion of the individuals are immune to the disease, especially through vaccination. The level of vaccination needed to achieve herd immunity is varies by disease. R naught actually indicates constant number of infected people. Below 1, that means transmission to fewer people, and above 1, transmission to higher numbers. So you see, according to WHO, COVID-19 reproduction number is understood to be between 2 to 2.5. It may also be as high as 5. So incubation period means the time between catching the virus and beginning to have symptoms of the disease. 14 days following exposure, with most cases occurring approximately 2 to 5 days after exposure. An infected person can transmit the virus during this period, that is in asymptomatic period. The main symptom of COVID-19 disease includes fever, coughing, shortness of breath, trouble breathing, fatigue, etc. The virus can lead to pneumonia, respiratory failure, septic shock, and death. Many COVID-19 complications may caused by a condition known as cytokine release syndrome or a cytokine storm. This is when an infection triggers immune system to flood bloodstream with inflammatory proteins called cytokines. They can kill tissue and damage organs. Coming to fatality rate. Fatality rate means the proportion of deaths a disease causes within a group of people who have the disease. Individual of any age can acquire this SARS-CoV-2 infection, but the fatality rate increases with the age and pre-existing medical conditions such as cardiovascular diseases and diabetes. Then period of infectivity, which means the interval during which an individual with COVID-19 disease is infectious, determined by evaluating viral RNA detection from respiratory and other specimens after symptom onset and later in the illness. It is currently uncertain for COVID-19 disease. Then seasonality of the virus, that is season or weather during which the virus remains most active. Coronaviruses in animals are not always seasonal but have historically been so in human for reasons that are not fully understood. There is, however, no evidence so far about the novel coronavirus seasonality. Coming to diagnosis of SARS-CoV-2. Here we have mentioned three methods: serological test, that is rapid antibody detection test; molecular test, that is reverse transcription polymerase chain reaction or RT-PCR; then pool testing method. In serological test, we use immunoglobulins, which are also known as antibodies. These are actually glycoprotein molecules produced by the plasma cells WBC. They act as a critical part of immune response by specifically recognizing and binding to particular antigens such as bacteria or viruses and aiding on their destruction. IgM is the first antibody that body makes when it fights a new infection, and it does not stay for long. While IgG can take time to form after an infection, thus indicating an old infection. If antibodies are present in the sample, this binds to the antigen immobilized on the test strip and give a colored reaction. Thus, there are blood, serum, plasma tests carried out to determine whether an individual's body has IgM and IgG antibodies developed against SARS-CoV-2. Coming to molecular test or RT-PCR, this identifies the presence of virus itself based on its genetic fingerprints. When a person is suspected to be suffering from COVID-19, an oral or nasal swab is taken. Then viral RNA is isolated from these swabs using a variety of chemicals. Enzymes are then added to transcribe the RNA into DNA. This DNA is put into real-time PCR machine that xeroxes the DNA, making thousands of copies of genetic material in the sample. Scientists then use a set of DNA fragments that complement fragments found in coronavirus. If any viral genetic material is present, these fragments will bind to it. Next is pool testing method. This was developed by German Red Cross Blood Donor Service in Frankfurt. It involves simultaneously testing a combined sample from multiple people from a household or a local cluster to widen reach and speed up results. It is suitable for expanded testing in larger population group. At this time, there are no specific vaccines or treatments for COVID-19. The most effective measure has been to isolate people who have tested positive, quarantine them, and increase social distancing to stop the spread of the virus. However, there are many ongoing clinical trials for evaluating potential treatments. Several studies are focused on an antiviral medication called remdesivir, which was created to fight Ebola. Chloroquine and hydroxychloroquine. These medications are approved to treat malaria and autoimmune conditions like rheumatoid arthritis and lupus. Studies on their use against COVID-19 have mixed results and research is ongoing. Next is ritonavir or lopinavir, which is licensed treatment for HIV/AIDS. Ritonavir or lopinavir with interferon beta 1A is the another one. Interferon beta 1A is a molecule involved in regulating inflammation in the body. Then convalescent plasma therapy. It is approved use of blood plasma from recovered patient to treat severely critical COVID-19 patient. There are more than 20 vaccines currently in development, with 2 in clinical trial phase involving human testing. Other than the 8 CEPI funded vaccines, there are also 14 other institutions, including 2 from India, that is Pune-based Serum Institute of India and Ahmedabad-based Zydus Cadila, which are engaged in developing vaccines for the infectious disease. Next is challenges in vaccine development in case of pandemics. In case of pandemic, it's a big challenge because as soon as a vaccine is approved, it is going to be needed in vast quantities. In pandemic situation, countries also have to compete with each other for medicines. The challenge is to make sure that the vaccine gets to all those who need it. And it's normal for a virus to change or mutate as it infects people. The SARS-CoV-2 is a RNA virus, so there is more chances of mutation. That's another big challenge in developing a vaccine. Vaccination is one of the major success stories of modern medicine, greatly reducing the incidence of infectious diseases. Here I have mentioned few of the type of vaccines, which are live attenuated vaccine, LAV, inactivated vaccines, then subunit or recombinant vaccines, conjugate vaccines, and RNA vaccines. Live attenuated vaccines contain a version of the virus that has been weakened in the laboratory so that it cannot cause disease. Because these vaccines are so similar to the natural infection, they help in preventing the virus by creating a strong, long-lasting immune response. Just 1 or 2 doses of most live vaccines are enough to give a lifetime protection against a germ or disease they cause. Inactivated vaccines. An inactivated vaccine or killed vaccine is a vaccine consisting of virus particles that have been grown in culture and then lost disease producing capacity. These vaccines usually do not provide immunity as strong as live vaccines, so needs several doses over time, which is called booster shots, to get immunity against a disease. Subunit or recombinant vaccines. These vaccines use only specific pieces of germ, that is the key part of the germ. For example, in case of viruses, generally capsid is considered. It can also be created by genetic engineering. A gene coding for a vaccine protein is inserted into another virus or into producer cells in culture, and when the carrier virus reproduces or when the producer cell metabolizes, then the vaccine protein is also created. When injected to an individual, the immune system recognizes and expressed proteins and provide future protection against the target virus. Then conjugate vaccine. A conjugate vaccine is a type of vaccine which combines a weak antigen with a strong antigen as a carrier so that the immune system has a stronger response to the weak antigen. This vaccine currently in use for children against pneumococcal bacterial infection. Then RNA vaccine. RNA vaccines work by introducing an mRNA sequence which is coded for a disease-specific antigen. Once produced within the body, the antigen is recognized by the immune system, preparing it to fight. RNA vaccines are faster and cheaper to produce than traditional vaccines, and an RNA vaccine is also safer for the patient as they do not produce by infectious agent. Most current research is into RNA vaccines for infectious diseases and cancer, but no vaccine made from RNA or DNA has been approved till date. Vaccine development involves the process of taking a new antigen or immunogen identified in the research process and developing this substance into a final vaccine that can be evaluated through preclinical and clinical studies to determine the safety and efficacy. Different phases involved in vaccine development process. The exploratory is research intensive phase of vaccine development is designed to identify natural or synthetic antigens that might help to prevent or treat a disease. After that, sequentially the phases includes preclinical, clinical development, then regulatory review and approval, manufacturing, and finally quality control. All these phases takes around 12 to 15 years in general, but as per need, due to this outbreak of COVID-19, the process of vaccine development has been fastened up by the researchers to save this world from COVID-19 disease. Few of these news that have been recently coming up includes the University of Oxford has developed a vaccine candidate where they uses a weakened strain of common cold virus, adenovirus, and is combined with the genetic material of SARS-CoV-2. This will enable the body to identify the spike protein of novel coronavirus. The vaccine candidate is now in clinical trial. Massachusetts-based biotech company Moderna is developing an RNA-based vaccine in collaboration with the National Institute of Allergy and Infectious Disease (NIAID). Chinese scientists are claiming to have successfully tested a potential vaccine for the novel coronavirus in monkeys. To conduct this experiment, the researchers injected the monkeys with the potential vaccine PICOV-VAC, which is made by Sinovac Biotech, a Chinese biopharmaceutical company. US-based Pfizer pharmaceutical company and its German partner BioNTech are working together on 4 RNA-based vaccine candidates. They also began clinical trials for their vaccine candidate named BNT162. Their vaccine candidate is based on specially designed messenger RNA that is similar to Moderna vaccine, and the trials for testing the vaccines are taking place in USA. As per recent reports, the Indian Council of Medical Research (ICMR) has teamed up with Bharat Biotech International Limited (BBIL) for developing COVID-19 vaccine. It is important to note that the vaccine will use the virus strain isolated at the National Institute of Virology that is situated in Pune. Moreover, the Serum Institute of India (SII), which is the world's largest maker of vaccines by volume, has partnered with Oxford University to produce to 60 million doses of potential vaccines. Inovio Pharmaceuticals, a biotechnology company, has developed a potential vaccine in its San Diego lab and is all set to begin phase 1 trial at the University of Pennsylvania. Now, how can you help stop the spread of coronavirus? Continue to follow safety practices such as wearing a cloth face mask in public places. Because the virus spreads from person to person, it's important to limit your contact with other people as much as possible. So take care of hand hygiene, then take care of respiratory hygiene, and social distancing during this pandemic is most important. It's a big question now how long the pandemic will continue. It depends on many things, including researchers' work to learn about the virus more and more, their search for a treatment and a vaccine, and the public's support and effort to slow the spread of the virus. Various vaccines are in different stages of development and testing. This process usually takes years. Researchers are speeding up the process as much as they can. The best way is we need to prevent the spread of the COVID-19 with prevention measures. With this quote, rise above the storm and you will find the sunshine. I am concluding this lecture here. I hope this information will be useful for everyone. Thank you. Stay home, stay safe.